Good morning, everyone. If morning is where your time zone is, welcome to the 6th annual ATTIS, the Annual Translation and Transformative Informatics Symposium, started in 2017. And the title this year is Explore More About the World of Genomic Informatics with Tutorials. So I'm James Cimino. I'm a professor of medicine and the director of the Informatics Institute here at University of Alabama Birmingham, UAB. And this conference is sponsored by the Informatics Institute, along with some others. I believe the Center for Clinical and Translational Sciences is co-sponsoring. So my job is to simply welcome you, kind of get the ball rolling. So let me do that. All right, so I was trying to think what to talk about because translational science is not my area of expertise. So what I did instead was I thought I would let the programs talk for themselves. And so I gathered up, thanks to Amanda Carballo, I got the programs from the first, all 6 of these conferences, and I ran them through WordMap. And the 2017 meeting was in person, and I don't actually have a attendee list or a lot of details about it, but I do have the flyer, and you can see the WordMap bioinformatics, a very top topic. And then you can see other themes actually came out very interestingly. So things like continuity, collaboration, networking opportunities, collaborative relationships. So, and in fact, the title was really about building relationships in a network for carrying out team science. And so that really, it was surprised, actually, pleasantly surprised to see that come out. Now you'll see another word here, chaos. And so one might be led to conclude that all of this collaboration and networking can cause chaos. Or you might conclude more favorably that chaos is the reason for establishing networking and opportunities and collaborations and so on. In fact, the real reason is that the first meeting was in person, was held at a place in the Lister Hill Center called the Edge of Chaos, which is this big open meeting space. And it is a little chaotic, but that's why the word chaos is in there. So 2018, also in person, we had 18 attendees and 27 speakers. And you can see you can see it, but I can't because I've got a Zoom thing hanging over it. And now we can see that bioinformatics was replaced with informatics as the leading topic. And bioinformatics is still there, but not as prominent, and at least relatively. And part of the reason is that there are other things modifying the word informatics, like clinical informatics, cancer informatics, pediatric informatics. So there were actually other domains than bioinformatics, which seems appropriate given that this is about translational science and not just bioinformatics. So 2019, we had a decrease in attendees. It was only 40 attendees, 10 speakers. It was a much shorter meeting. But now you can see genetics has gotten into there. Informatics is still big. Institute, I should have removed that word from the list. That's, I'm sure, referring to the Informatics Institute, maybe some other institutes. Rheumatology, though, is now there. Immunology, so we're seeing some relationship to things like immunology, autoimmune disease, and we're seeing people's names there. And we see the word Hilton. And it wasn't about the Hilton or Chelsea Hilton or anything like that. It was really that the meeting was held at the Hilton because it was in conjunction with the Mid-South Computational Biology and Bioinformatics Conference, MC Bios. And so that's why it was a short meeting, because it was only half a day held in conjunction. We hosted that meeting that year. So that was the last in-person meeting we had for in this conference. 2020, you remember, was the start of the pandemic. So we switched very quickly to a virtual meeting. We had 68 attendees, despite that, online attendees. We had 19 speakers. And hold on, I got to move my little Zoom box so I can read the word map. Informatics, again, big word. Institute, sorry, forgot to remove that one. But now we see microbiology as a topic. And we see UBright, which is our platform for biomedical informatics research technology enhancement. And this meeting then was virtual, although we did have lunch. We had already bought lunches. So people came and picked up their lunches, but at a distance, wearing masks, hand sanitizer, everybody wearing gloves as we handed out the sandwiches. So those of you who picked up sandwich may remember that. Okay, last year we definitely knew we were going to be virtual, and we advertised it as such. And now we had 119 attendees, 24 speakers, and we could see genetics now, much bigger prominence there. And we can see precision, and we can see platform. So talking about ways of carrying out analysis. And now this year, we have 129 attendees, so the largest that we've had, 25 speakers, again, largest number that we've had. Whoops. And now we see medicine in there, interestingly. Whoops. See what else we got. Sorry about that. So medicine, which is really what all this is about, right? I mean, we're not trying to figure out how to cure fruit flies and zebrafish. We're really trying to figure out how to improve human health and reduce disease burden. Okay, so the speakers, 25 speakers, a lot of them are from Alabama, as you might expect, several institutions in Alabama, but you can see we have people from Louisiana, Texas, California, Indiana, Missouri, Maine, and Washington. So we've got a pretty good spread. No international speakers this year, but a pretty good national representation. And so I just want to conclude by saying thanks to the program chairs, Jake Chen and Chen Chang, who did a tremendous amount of work putting this all together, figuring out who, inviting people, getting the program put together, coercing people to be session chairs and lead the discussions. Staff, Amanda Carballo, Ryan McCain, Nafisa Sani, and also other folks in the Informatics Institute provided a lot of support. And then also I want to thank the moderators and speakers, because of course we couldn't do this without you. And there's 25 of you, and it's great to have you here. And so I just wanted to say my sincere thanks for helping us put this meeting together, but especially Jake and Chen, who really labored hard. We've got to find some other people to chair this meeting besides them. Alex Rosenberg did it once, I believe, but Jake and Chen have carried the bulk of this. So we'll find somebody, train somebody up for next year. And then finally, I want to remind you that at the end of the meeting ends at 5. At 5:30, there'll be a reception, and this will be in person, Shelby Research Building 105. And we will have some pretty good food catered. So there's the menu, but I just wanted to pique your interest and whet your appetite. And this is, this will definitely take the place of dinner. It might be better. It looks better than the dinner I was planning for myself. So hopefully we'll see those of you who are at UAB can stop by. We've got plenty for everybody. And so with that, I will relinquish command and over to, I guess, Chen.
Thank you, Jim. Thanks for the nice introduction. So I would mention, I'll advertise a little bit. So we have 2 excellent keynote talks, and we have 2 technical talk sessions. One in the morning, just after this remarks, and the other is in the afternoon. And we have a career development session in the noon lunch area, and after that we have a trainee flash talk session. So we have trainees from nationwide. So make sure you can stay the whole day, then you will enjoy this meeting. And yeah, we have 2 tutorials after the talk, the technical talk too. So enjoy, and thanks for coming. Elliot, probably you can.
Right, I'll take over.
Thank you.
Thanks, Chen, and thanks, Jim, for your welcoming remarks. So I'm hosting the first technical talk session of the 2022 ATTIS. Each speaker will have 20 minutes, so I think roughly 15 minutes to talk, and then 5 minutes for questions. And I'll have a little timer going to remind you at the 15-minute mark. So let's start. The first talk will be Ryan Miller, Director of the Division of Neuropathology in the Department of Pathology at UAB, talking about probing the glioblastoma kinome. Ryan, take over, please.
Thank you. I'm honored to be presenting to you today, and I really appreciate the opportunity to kick off this session. As Elliot said, I'm the Director of Neuropathology here at UAB, and my focus is on gliomas, particularly glioblastoma. So these are malignant brain tumors. that diffusely invade the normal brain. They're very severe diseases. As you can see here, gliomas constitute 80% of primary malignant brain tumors. And of those tumors, glioblastoma is the most common. Unfortunately, we have no treatment options, few treatment options for these tumors, and therapeutic decisions really remain empirical. This shows data from a landmark 2005 study that established combined radiotherapy and temozolomide as the standard of care for this disease. And our therapeutic armamentarium is pretty paltry. We really only have 3 drugs that have been approved for any indication in gliomas, and temozolomide is really the only one that's proven to extend survival. So when we ask what the problem is with this limited therapeutic armamentarium, we look at clinical trials, and 95% of clinical trials for oncology indications wind up failing. Most of those fail at the phase 2 stage, where efficacy is tested. And this really emphasizes the need for better patient stratification, which in today's precision medicine era really means molecular diagnostics, as well as better preclinical modeling, so that we reduce the denominator and therefore reduce attrition in clinical trials. So glioblastoma was the first tumor that was deeply genomically characterized by the Cancer Genome Atlas, and I had the good fortune to be involved in that effort. And what we found was that these tumors had recurrent genetic alterations, not necessarily in specific genes, but in pathways. And so, as you can see here, there are 3 what we call core pathways that are mutated in this disease. And those include the receptor tyrosine kinases, the PI3 signaling pathway, and the G1S cell cycle control pathway, highlighted by CDKN2A and RB. So we were particularly interested in the kinases such as EGFR, which is the most frequent dominant oncogenic mutation in GBM. And as you can see here, that's a very frequent event in this disease. It constitutes between 50 and 60% of all glioblastomas. So if precision medicine is going to work for this disease, EGFR is an excellent target. So we wanted to specifically look at one mutation that constitutes about half of EGF altered glioblastomas, and that's N-terminal deletion mutation that's called EGFRV3. And it is the most common oncogenic mutation in GBM. And as you can see from this oncoprint from CBioPortal data, frequently co-occurs with CDKN2A, loss of function mutations, most commonly homozygous deletion, as well as P10 deletion, which is a negative regulator of the PI3 kinase pathway. So we wanted to investigate small molecule inhibitors of EGFR. Now, the initial enthusiasm for this approach was dampened somewhat by a series of failed clinical trials in the mid-2000s. And the reasons for those failures were that the drugs tested didn't get into the brain. So the brain has a blood-brain barrier that prevents small molecules from crossing. So really the wrong drugs were tested. And we didn't use molecular diagnostics to enrich the patient population for those tumors that had EGFR abnormalities. And then a third theoretical reason for their failure is acquired resistance. And that's particularly what my laboratory is interested in. As you can see, a number of EGFR inhibitors have been tested in the clinic in this context. But you'll also note that most of them have very poor blood-brain barrier penetrance. So we wanted to develop a series of preclinical models where we can investigate the mechanisms of EGFR tyrosine kinase resistance. And in particular, we wanted to use a multi-omic approach to identify differentially expressed kinases by RNA sequencing and identify on the protein level those kinases that were differentially expressed using a proteomics, a mass spectrometry-based proteomics technique that I'll tell you about in just a minute, called multiplex inhibitor bead mass spectrometry. We then wanted to do either genetic or pharmacologic validation of those altered kinases, and then ultimately design a dual kinase inhibition strategy to treat this disease. To do so, we utilized a series of genetically engineered mouse astrocytes that all had CDKN2A deletion with or without EGFRV3, abbreviated here EV3, and with and without PTEN. So we had all of the genetic combinations required to dissect not only the oncogenic effects of EGFR plus minus PTEN, but look at their response to therapeutics as well. And this just shows a Western blot that validates that our genotypes are accurate. This is also validation for EGFR and PTEN. This is using RNA sequencing data, and again shows that we have the appropriate genotypes in our models. The biology of each of these models is different. So as you can see here from, this is in vitro proliferation data. The doubling times are greatly accelerated when you overexpress EGFR-V3. And it really doesn't change much when you add PTEN, but these tumors become much more lethal when injected into the mouse brain. And these tumor cells also increase their self-renewal by extreme limiting dilution analysis. So our molecular credentialing pipeline really involved RNA sequencing and MIB-MS-based kinome profiling. And I'm not really going to talk about epigenetics. And since these are genetically engineered mouse astrocytes, we really didn't need whole exomes sequencing on those. Okay, so here's the promised data about MIB-MS. Basically what it is a series of broad spectrum kinase inhibitors that are chemically conjugated onto cephalose beads and layered in a column format. So this is an enrichment type of approach where we can probe, as you can see here in this panel C, the entire kinome tree. And we can change the composition of the MIB enrichment strategy to target specific kinases within different subfamilies of the entire kinome. So we have previously published on this, and as you can see here, this is a series of mouse astrocytes that were treated with a PI3 kinase inhibitor over an acute time course of 0 to 48 hours. You can see the kinome here at the protein level is very dynamic, and a series of kinases, or a subset of kinases, become inhibited with a targeted inhibitor. Some get reactivated, and then some are what we call alternatively activated. And it turns out when you inhibit the PI3 kinase pathway, the MAP kinase pathway goes up, and that represents a target for dual kinase inhibition. So given the fact that we had a series of genetically engineered mouse astrocytes, we wanted to ask the question, what do their transcriptomes look like? And as you can see from this from this PCA plot, these mouse astrocytes, the first principal component largely separates them based on EGFR status, and the second principal component separates them on the basis of PTEN. And you can see this reflected also in the hierarchical clustering. This extends to the kinome. So here is a subset of differentially expressed kinases that basically perfectly separate the mouse astrocytes on the basis of their genotype, specifically EGFR and PTEN status. This also extends to the protein level. When we look at the multiplex inhibitor B mass spectrometry data, there's a little bit less separation on the PCA, and the hierarchical clustering is a little bit fuzzier, but the kinomes are still distinct on the protein level. So we next wanted to identify kinases that tracked with EGFR status. And to do so, we did pairwise comparison between our CEV3 model and the model that lacked EGFR V3, so we call it C. And as you can see here, the major kinase that's different is human EGFR because it's engineered to be overexpressed. And this correlates significantly with mRNA expression. So then we looked at that same pairwise comparison in 4 different data sets on the RNA level, where cells had either been cultured with serum or starved, as well as the protein level in cells that had been cultured with serum or starved. And what we identified only at the protein level was a series of kinases listed here that were highlighted by CDK6. And if you do volcano plots or MA plots, you can see that CDK6 tracked with EGFR status in this particular comparison. So then we wanted to pharmacologically validate this as a viable second target for combinatorial therapeutics. And there's a number of FDA-approved kinase inhibitors that target CDK6, one with exquisite brain penetrance, that's abemaciclib. And so we asked the question, could we develop a therapeutic strategy that targeted not only EGFR, but CDK6? And as you can see here, on the protein level, but not on the mRNA level, we found increased expression of CDK6 in our cells that had EGFR overexpression and wild type PTEN. And this was verified by Western blot. So then we wanted to ask the question, if we gave a combinatorial therapeutic strategy to mice bearing these EGFR B3 driven tumors, will we get a survival benefit? And that is exactly what happened. And we only got a survival benefit with the combination therapy. Neither the drug targeting EGFR, neratinib, or abemaciclib, which targets CDK4 and 6, neither one of those alone were effective, but only the combination was effective. And we've since extended the treatment duration of these mice and get and see the same thing. So with that, I want to acknowledge my lab, our collaborators at UNC, TGen, Mayo Clinic, UCSD, University of Minnesota, and UCLA, and thank the funding agencies that provided the funds to do this work. And I'll take any questions.
Great, thanks, Ryan. If anybody has any questions, please, if you want to speak up or if you want to type your question into the chat, I can relay it for you. So questions.
So excellent work, Ryan. I have a quick question regarding the kinase. So it's a mass spectrum, right? It's the proteomics kind of work, right? Have you tried to compare with your profiling results versus the TCGA results? I think TCGA has some RPPAs or even transcriptome data. What's the yeah, the level consistency or
You mean what's the consistency between
or
MIB-MS
if
and
yeah,
RPPA?
if they are comparable or
Yeah,
some
so
dramatic difference. Yeah.
the reason why we wanted to go a mass spectrometry-based approach is that techniques like reverse phase protein arrays require reagents to probe specific kinases. So you really need antibodies. And ideally, you need antibodies to phosphoepitopes. So what the MIB-MS approach provides is an unbiased way to look at the entire kinome, because we pull down active kinases. So all of these drugs are ATP pocket binders. And so we pull down active kinases and measure their expression with mass spectrometry. So it's really a complementary technique. It provides basically expression data specifically of active conformation kinases. So it's a complement to RPPA.
May I have a quick question?
Thanks, you.
Yeah. So in the last couple of slides, you mentioned that there are additional target you identify in addition to CDK6. There's the other SYK and LAC2 and other PIP. So you think that you are going to test some kind of combination of those inhibitors and see how that works?
Yeah, so the idea would be, assuming we're currently validating these results in patient-derived xenografts that have the same genetic mutations as our mouse astrocytes, and assuming we get similar data in those model systems, we can envision a clinical trial where we would do molecular diagnostics for CDKN2A, EGFR, and PTEN, and we would give a combinatorial therapy only to those patients that have EGFR mutations and wild type PTEN. Because what I didn't show you is that our model with PTEN deletion and EGFR V3, we saw no synergy in vitro, and we saw no survival advantage in vivo.
Thanks.
Mm-hmm. Any other questions? Okay, thanks, Ryan, for a great presentation. So next up, we have Jake Chen, who's a professor of genetics and computational science and co-director for bioinformatics of the Informatics Institute. Jake will be talking to us about the statistical enrichment analysis of sample clinical attributes using either SEAS or SEAS. And Jake will tell us how he pronounces the acronym. Jake.
Yeah, well, thank you. So this is a very specific piece of work that we published last fall, and it does pronounce as a SEAS, which is an acronym for SEAS, Statistical Enrichment Analysis of Samples. If you're interested in the details, and here's the paper, and I want to just go through this presentation to show you what it does and what the type of problem that we're facing that's benefited from this new tool. So a little bit of a background. In functional genomics or genomics, there's a lot of the exploratory analysis work that's going on. The most typical one is this sample matrix. If you look at this matrix, on one axis is gene, the other axis is samples. And in the matrix, we'll fill it with values of gene expression. And then this is a functional genomics gene expression profile. So a lot of time we use R, we don't care about the X and Y annotation until at the later stage. And when you think about it, we really care about the dimension of genes. for many years, and we've published tools, and we talk about the downstream analysis and really focus on how do you annotate these genes that are determined to be significant from this matrix. And then we can use pathways, networks, and this is outside of the scope of today's talk. But on the other hand, there's a lot of sample annotation. If we're dealing with human samples, and the amount of annotation information can also be very large, not just limited to sample ID. For example, in this, I show the patient ID, age, survival information, chemotherapy information. In large databases such as the TCGA for cancer, some of the sample may have up to 200 or 300 clinical attributes. And there's actually a need to actually also analyze this information. So one way to look at this matrix and explore it very traditionally is to use a clustering approach. You can do clustering on the sample side, clustering on the gene side. And this is one example of using different type of a clustering algorithm to arrive at some kind of an insight on the sample. And clearly, of course, when you try to use a different clustering algorithm, in this example, there's going to be some clusters that's very similar, and then some cluster is going to be dissimilar depending on the clustering algorithm. When you're choosing the correlative clustering method, and then that difference can be a little bit higher. So clustering is not very stable, and I think researchers are always looking for a more stable way to really look at the relationship between samples. So one of the common practice now expanded by new just a machine learning method is called a dimensionality reduction techniques or embeddings. I think for many years, and the principal component analysis, which is a linear embedding, has been predominant. And then the other nonlinear method, especially ATTIS and UMAP, has become popular because of the single cell analysis. But few people have actually looked at their application and still coming back to the bulk analysis of patient samples, right? So in single cell analysis, it would be understood that there's a lot of heterogeneity of cells. And if you can actually capture more of a local heterogeneity with some aspect of a global distance, you can actually do very good clustering based on the embedding, right? The linear techniques is not very well. Actually, this example from this previous publication shows that as to analyzing the heterogeneous patient samples in bulk, linear embedding also do not separate samples very well. They tend to cluster the samples, while nonlinear embedding can do a little bit better. Especially in this example, there are two batches, batch 1 and 2, and the UMAP can actually show the different batches quite well, whereas some of the other earlier nonlinear embedding techniques do not show very well. So one of the inspirations, as I mentioned earlier, is that we want to actually explore clinical attributes using embeddings. And this is one of the motivational story by another paper. And this is recently published actually in Cell Reports. And in this example, and they have the smoking status labeled across the sample. So they're current smoker, former smoker, and never smoker. So no matter what you use to label the samples, the bulk RNA-seq samples, and they seem to be, they're all intertwined. But when you switch to another clinical attribute, gender, and there seems to be some trend that in the middle section, there seems to be an enrichment of males, right? And the paper actually further on to use this clinical attribute, which is actually the gender itself, and do a clustering C1, C2, and C3. And they have determined that in C1, which is this region, there's an enrichment of female, and then in this region, enrichment of male and a male. So there are clearly a separation by gender according to this a simple clinical attribute. And from this point on, if you just further do the heat map analysis, you can gain very good insight of the correlation between gene signatures here and the cluster C3, which is mostly enriched by male, and another cluster at the gene level, which is enriched by samples of C2, which are female. So essentially, what they're trying to say is that the gene signatures, according to this study between male and female, can be quite different when you employ some clinical analysis early on into the functional genomics analysis. So this kind of exploratory analysis could not have been actually automated by your differential gene expression analysis, or probably would be a much weaker if you defer it until the later stage. So I think our motivation is we want to actually do this repetitively and do it for 200 clinical attributes, what we call the clinotypes of a selected sample size, such that we can zoom in to particular sample group in the embedding and get which clinical attributes or clinotype are enriched against the background. Hopefully the software tool can screen through hundreds of clinotype and tell us which one is the one. So in C's, we start with data input and essentially the sample clinical data plus another file that's embedding data. If you don't provide the embedding data, the C's will try to learn the embedding and generate the embedding from the original sample gene expression level data. If you do provide it, then it will take use of it. And then it would create visualization, and you can explore it. You can actually identify a subcohort. A subcohort essentially is a cluster or selected samples, whichever way that you define it, such that you want to actually select it as a focus. And then once we have that, we compare the focus against the background using something that is very similar to the gene set enrichment analysis, hence the term statistical enrichment of samples. And then we'll generate the final result. So here's a illustration how it works. In this coordinate X and Y, that's embedding. Suppose that you use some kind of embedding, and then you can select a patient of interest and then define a oval radius. And then the calculation of statistics become fairly straightforward. So you have this total number of samples in the population, Small n is samples in the selected cohort, and the big K is the total number of samples with a specific clinical type in the population, and small k is in the selected one. And then you plug in this hypergeometric test, and you can generate with the software automatically something like this. Here's your clinical attributes. There's a p-value, then there's just a p-value, whether it's enriched or not. And then we show it in the software, right? This is a snapshot view. So in this view, we intentionally did not actually just select one neighborhood, but selected some in this neighborhood, some in other places. And that selection can be completely done according to labels, or it could be done according to the region. In this case, we choose the labels. And then the software would say what sample you selected, and then it would report the sample characteristics. And then the enrichment effect. So with a tool like this, it seems pretty simple. Then what are the potential applications, right? And I want to actually show some example applications. Just as in the previous talk, and we a lot of you learned about the glioblastoma, and we actually collaborated with UAB Neuro-Oncology group, specifically Dr. Chris Willey. He has actually sequenced several patient-derived xenograft models, or PDX. One of the things that is he doesn't have a lot of them. So in this embedding, you see everything that's colored either green or the blue are the PDX samples. And the one that's actually coming from the TCGA are colored red. So most of them are from TCGA, which is not the sequenced sample here at UAB, but we try to actually learn the labels to assign it to the patient-derived xenograft models based on the label information, especially the rich amount of information, clinical information from TCGA. So for this particular sample, X1516, and so you can see that the before and after radiation treatment, it does not move at all. In fact, the fact that it doesn't move, and then you can infer that the label is more or less the same. That implies it's a radiation resistant, and it is rightfully so. But when we look at another example, X1153, it actually moved. Originally, the parental line is in here. And afterwards, actually the label should be here. So afterwards, and we'll look at the neighborhood, it moved to somewhere in the center. When we apply C's analysis and we try to see what are the days of survival before and after, and we got 242 on the samples on average and 324 afterwards. So all of a sudden you can actually realize that, oh, by applying the analysis such as C's, and you can actually pretty much learn the labels and even correctly predict without doing experiment that this line should be resistant, because the neighborhood also, it's a sensitive, sorry. So because the neighborhood become better, the survival become better. So that means that its radiation really help. So another way to do this is that we're trying to combine the C's analysis with the gene terrain, which I wouldn't actually talk about detailed, but it's you can think of it as a fancier heat map analysis that was conventionally done. So here we have another embedding, and the label here, actually there are 4 subtypes of GBM: classical, mesenchymal, neural, per neuron. And we look at one neighborhood of samples. We actually generate a heat map according to the gene terrain algorithm. And this is what the gene terrain algorithm look like. And when we go to another area that is actually predominantly per neural samples, we get another gene terrain image. And these are relatively stable and distinct patterns. And from these, you can actually get the C's to tell you what are the clinical attributes that look like. So here's a figure that we showed that by patient age, as a clinical attribute alone, the group 1 is actually a lot less than people who are less than 50 years of age. And then group 2 is much older. So, or is that the other way around? Yeah, no, I think group 2 is a lot more people who are younger than 50 years of age. Group 1 is actually shifting toward the older. And also at the gene signature level from the gene terrain, and I wouldn't actually use for this talk, but there's a lot of a follow-up. You can find exactly what is the gene network and gene signature underneath these this particular area, which in one subtype of a GBM is all downregulated, whereas in the per neural are all upregulated. So I think we're excited that this tool is was made, and we're continue to improve the usability. You can go to the website, like and there's a web server aimed dash lab at the shiny apps.io/Cs, where you can actually go check out our manuscript, which also contain these links. So with that, and I want to thank Sam Bardi, who will be joining as a PhD student from India, and she did a lot of the engineering, and my group actually participated in the design and evaluation of the software. Chris Willie and Christian Stackhouse, who also contributed biological insight to this work. Thank you very much. I look forward to some maybe questions.
Thanks, Jake. I think we have time for one question. Someone want to either speak up or type their question into the chat? One question I had, Jake, I'm not sure I completely understand the concept of sample embedding. Is that just allowing you to focus in on a particular clinotype, or do I have that wrong?
Yeah, the sample embedding allows you to look at the relatively similarity relationship of samples in a particular area. So traditionally, if you don't do this, your view of the sample relationship is quite linear according to the clustering. And so you can follow the relationship through the tree structure, and it's also not very stable. So the idea of embedding is it will capture all of the high dimensional relationship and try to project it in a low dimensional space, preserving the similarity the way that it should be in the high dimensional space. And it's also better than the linear. So it's there's a lot of a complex mathematics theory behind it to make sure that this kind of embedding are representing the true relationship that captures some local characteristic. So it's very mouthful, but I think you just have to trust that this is a trend that people use it more and more as opposed to clustering.
Okay, thanks, Jake. I think we need to move on at this point. So our next talk is by Mark Chaisson, who is an assistant professor of quantitative and computational biology at the University of Southern California. Mark will be talking to us about the motif composition of variable number tandem repeats impacts gene expression. Mark, please take over.
Thanks, Elliot. Can you give me thumbs up? I'm using a slightly different audio setup, so if you hear me fine, thumbs up. If not, I'll quickly change.
It looks good.
Okay, cool. Okay, yeah, and thanks to you, Chen, for inviting me here. So let's start this. Okay, so we'll just kind of dive into some recent results of a consortia that our lab is part of, is the Human Genome Structural Variation Consortium. And this is a consortium that has been dedicated to essentially sequencing, assembling in a haplotype resolved manner, and using a set of diverse genomes, and using those diverse genomes to detect structural variation. So the problem of finding structural variation from these assemblies is a pretty straightforward task. You can assemble them, they have very high contiguity, N50s approaching 20 megabases, and you find essentially comprehensive discovery of structural variation from haplotype resolved assemblies. And one of the kind of initial observations that I had in a pilot study that's confirmed by this study is that the majority of structural variants by count that are discovered in this data set land in about 4.6% of the genome. And that 4.6% is composed of sequences that are called variable number tandem repeats. So if kind of breaking that down, about 2/3 of inserted sequences in any given human genome, and about half, a bit more than half, of deleted structural variant sequences. I should quantify. A structural variant is a gain or loss of DNA that's at least 50 bases. And so just by the numbers, these are happening in a highly enriched fashion in these variable number tandem repeat sequences. So let's look a little bit about what the structure of these sequences look like. So, so it's about 3 to 4% of the human genome, and they're, pardon me, these sequences are small loci that are composed of repeated sequences of at least 6 bases. And so it's essentially a repeat motif that's repeated over and over in tandem. On average, it's about 215 bases per locus, but up to thousands of bases. And one key observation, so what's shown here is a dot plot of a VNTR sequence against itself, where every dot shows a shared sequence between one position and another position on the VNTR. And if it were a perfectly repeated sequence, you would see kind of like an exactly repeated checkerboard pattern. But what you see is this kind of changing pattern across the span of the VNTR sequence. And what this shows is that the composition of the VNTR, the motifs that are repeated, actually change throughout the particular locus that we're looking at. So they're highly variable. We find a lot of structural variance in these sequences. So they change in length between individuals. But when we look at the actual assemblies themselves, we can see that the composition of these variable number tandem repeats is also different between individuals. So for the sort of duration of a lot of the span that we've been studying variable number tandem repeats, it's been the discoveries in terms of the biological impact of this variant have been related or have been restricted to a few examples of clinical sort of known tandem repeat expansions. disorders. And then a few new discoveries, such as an intronic repeat of a VNTR in the gene called ACAN was shown to be associated with height in a group of Icelandic individuals. Furthermore, so that VNTR that I showed you before was an intronic that I showed the dot plot of, is a VNTR in a gene called CACNA1C that was shown a few years ago to associate with schizophrenia. And so one thing that's key is that most associations of tandem repeat disorders associate an expansion of a tandem repeat with a particular disease. This study was a change in what people observed because it actually associated composition of tandem repeats, or this particular tandem repeat, with an increased likelihood of developing schizophrenia. So this was independent of an association that was independent of length variation in this particular locus. But focusing on length variations, over the past few years, a bunch of different studies have looked at mapping GTEx data into either human genomes or human pangenomes, which I'm going to talk about today, and using specialized methods to associate tandem repeat length with nearby gene expression. Okay, so why are specialized methods needed to analyze VNTR variation in human data? So if we look at short read data mapped back to the human genome, what we can see is that the alignments are human genome over a variable number tandem repeat. The alignment coverage is highly non-uniform, and you see a lot of low mapping quality. So it's very difficult to just use standard variant discovery methods in repetitive sequences with short read data. So one way around that is to try and improve the reference that we map sequences to. And so to build those references, there's these consortia, Human Pangenome Reference Consortium, the Human Genome Structural Variation Consortium, that are doing long read sequencing and developing very high quality haplotype resolved assemblies of diverse sets of human genomes. With those haplotype resolved assemblies, people are developing methods to essentially merge all of the different sequences from multiple individuals into one representative reference data structure. So I say data structure because we're not considering the human genome to be a linear reference, linear sequence anymore. We consider it to be a graph, where any node in the graph represents a sequence that's shared possibly between multiple individuals, and different paths through this graph represent the different possible sequences of human genomes that one might observe in a population. So looking at, and what's shown here is kind of an overview of the main method that the Human Pangenome Reference Consortium is using to build up pangenome graphs. It was developed by Hung Lee, and it's essentially a multiple sequence aligner of large assembly, large haplotype resolved assemblies. So let's focus on what VNTR sequences look like in terms of their sequence composition, and how that might affect pangenome graph construction, and what we can do for pangenome graphs in VNTR sequences. So a pangenome graph is essentially taking a reference sequence. What's shown on the left is a set of a reference and a set of kind of cartoon examples of VNTRs. And a pangenome is essentially a multiple sequence alignment where any two sequences that are aligned are kind of merged to each other. And so this is sort of they often generate fairly messy structures in VNTR sequences. And importantly, there's multiple different alignments that essentially have the same score that can generate different graph structures for pangenome graphs in repetitive sequences of DNA. So this can cause some difficulties in terms of genotyping, alignment, and comparison between individuals, and we want a better representation of pangenomes in repetitive DNA. So this problem actually was studied a long time ago, including by Degui Zhi, who's going to be presenting later, showing approaches for repeat classification. So building what's called a repeat graph, where Similar to the pangenome graph, where every sequence is, where every node is a sequence shared among individuals, a repeat graph is a sequence that's shared among different positions in the same genome or different genomes. So it's essentially collapsing within the same and across genomes. So what we did to try and improve how we align sequences into variable number tandem repeats is a very simple pangenome graph. Essentially, it's just a glorified De Bruijn graph, where if we have a repetitive sequence, we essentially just collapse all of the repeats of a fixed length into the same node, and then repeated sequences correspond to cycles in this graph. And then if we add a new sequence that, say, has a different motif in it, that corresponds to a different set of branches and cycles in this graph. And so we developed a pipeline called Danbing TK, and what that does is it finds orthologous loci, VNTR loci between assemblies, makes separate VNTR or separate pangenome graphs, which are just De Bruijn graphs of these individual loci, and then has extra modules to map Illumina reads into these loci and infer variation. So on 64 haplotype resolved assemblies, we have about 85,000 loci that we detect variation in. The size of the graph, so the cumulative number of vertices that we observe, kind of strictly increases as we keep adding more and more genomes. And what's important is if we map reads back onto these loci, the total number of reads mapped per locus actually correlates very strongly with the length of the original locus. So if we want to do something like estimate VNTR length, we can map them back into these individual De Bruijn graphs, sum up the reads aligning into the graph, and then estimate the length of the VNTR sequence. So we did this starting a couple of years ago, and what we could show is that if we take whole genome sequencing from the GTEx genomes and map it into these De Bruijn graphs, we can discover a new set of EQTLs that have been missing from previous studies. Importantly, what we can show is that when you map sequences from different populations, they can map differentially to various cycles in the graph. So that means that different populations have different motif usages. And we thought that maybe it's possible that the different motif usages are responsible for changes in expression rather than just different lengths. And so before, what we did was to map sequences into these De Bruijn graphs and estimate length as kind of a total depth among all vertices within each graph. And we just do a simple modification where we only look at alignments to non-branching edges in this graph, where we kind of separate out what was this whole signal to signal that's kind of in separate paths in this graph, where each path corresponds to essentially a repeated motif in the NTR sequence. And if we map reads into this and we find a cis association between total number of reads mapping to a path and a nearby gene, we call that an E motif, where observation of changes in that motif are found to correlate with gene expression. So these motifs map really well. We're able to estimate the copy number of motifs with pretty high correlation to the ground truth in the underlying graphs. And just like before, when we looked for correlation between length and gene expression, there's widespread cis association between motif variance and gene nearby gene expression. And to the extent that it's actually in 2 orders of magnitude greater number of associations that are discovered for motif variation and gene expression. So in this data set, so the previous result that I showed about 300 E VNTRs, we have a new graph that's able to find association, about 4-fold increase in the number of VNTR loci where total length is associated with nearby expression. But when we look at motifs, it's about 20,000 motifs that are associated with nearby gene expression. So this kind of worried me, thinking that, okay, one of 2 things is happening. We have a lot of false positive variance, or we just have a bunch of variants that are being tagged by nearby SNPs in general regions that are in LD with motifs that actually, or variants that modulate gene expression. So we did fine mapping, and we're able to show that there is, in fact, a large number of E motifs. These are just short motifs that are associated with nearby gene expression that are actually don't have any sort of length that's associated with nearby gene expression. So, and importantly, 95% of E motif variants are in loci that don't have an association with only length with nearby gene expression. So just sort of wrapping things up. So one example that we looked at is the CACNA1C locus that I mentioned before, which was shown to have certain motifs that associated with nearby gene expression. When we look at the motifs that we map in this, in the De Bruijn graph that we created for this sequence, we're able to replicate a large number of the risk alleles that were identified for schizophrenia as alleles that are associated with a decrease in expression of the CACNA1C gene. We're also able to find one additional risk motif that wasn't discovered in the schizophrenia specific study. And so, so it's a short story, but I think that this is something that we're interested in applying to larger data sets to see if not only are there cis associations with expression, but there's also potentially new disease associations that can be found with this approach. So I want to say thanks to everyone in the Chaisson lab, and thanks to all the data generated by the HGSVC and the HPRC. And I'm happy to take any questions.
Thank you, Mark. Questions?
Mark, this is excellent work. So I wonder, the graph is based on the De Bruijn graph is for short reads or long reads? It's
right,
for
so
short
the
reads.
graph itself is made from the assemblies that are generated. So essentially, what we do is for every, so we annotate the boundaries of every VNTR sequence in, say, HD38. And then we find the orthologous map boundaries of the VNTR sequences in all of the different assemblies. So now we know roughly what sequences in each assembly corresponds to that VNTR locus in HD38. And then we cut all of these sequences out and form a De Bruijn graph on that. And essentially we have, for every VNTR locus, we have a disjoint De Bruijn graph. So this is from our first paper where we had about 30,000 different VNTR loci that we considered. Now we're up to about 85,000 VNTR loci by virtue of having higher quality assemblies and more assemblies, we're able to include more loci in our graph.
So could this graph be used for like a discovery purpose? So this kind of like typing, right? You know the haplotypes and then try to pinpoint to the region and then.
Well, so it is discovery in the sense that it's not, so a lot of the pangenome graph genotyping approaches look for sequences that kind of confirm a branch in the graph. And so what we're looking for, our discovery is essentially what is the overall depth of sequences in these graphs. So we can have new samples that have different lengths or different counts of composition, sequence composition. Rare variants will be missing in these graphs, and it's kind of one extension that I'm hoping the student will solve this summer, which is essentially to take pretty large UK biobank sized Illumina data sets, and we can do at least some rare variant discovery in these graphs.
That's awesome. Another quick question is that so the VNTR is on average several hundred kilobase pairs, several hundred base pairs, right? Or a few thousand. So it's still it could be within the hi-fi arrays or the CLR probably error, too many errors. But for hi-fi arrays, it can contain the VNTR entirely, right, in some of the rays. So how about the mapping-based approach to discover the VNTR?
Right. So we have a separate. So discovering variants in these sequences is, once you've assembled the genome, is a solved problem. So we have an aligner that can align whole genome sequence, whole genome assemblies. Everything aligns perfectly fine across these sequences. Newer nanopore data, it spans all of these sequences, and so alignments just have the variants in them. It's not a difficult problem to find. Classifying the variants, once you've found them, is actually a separate project that we have for long read data. Actually, I'll show on an HGSVC call in a couple of weeks. But in terms of finding them, it's more or less a solved problem.
Got you. Great, thank you.
Mark, are there VNTRs that are conserved both in terms of sequence and or location across species?
Yeah, so that's a good question. So we have another project where we essentially were tabulating, let's see if I can unhide an image. Essentially tabulating the motif composition or the repeat composition in various assemblies. And so we're building this up for all the human assemblies that we have, and then we tack on all of the primate assemblies that we have. And so what's interesting is that there are a lot of conserved VNTRs from primate assemblies, but there's also kind of newly born VNTR sequences in every different genome. So there's certain loci where it's only a few bases in one species and hundreds or thousands of bases in other species. So this was actually a project that I kind of started when I was a postdoc, and a different postdoc finished it in one particular publication, essentially tracking the birth of a few thousand VNTRs in human sequences. There's a slight enrichment in humans for VNTR sequences that may have an effect on regulation of neuronal genes. In terms of what the story is in primates, it's kind of an unknown thing, but we have a collaboration going on with Omer Gakumen to go and kind of classify what these variant sequences look like in at least non-human primate genomes.
Okay, thanks, Mark. All right, to round out this session for today, now we have Degui Zhi, who's an associate professor and director of the Center for AI and Genome Informatics at the University of Texas Health Science Center at Houston, to talk to us about informatics to unleash the power of biomedical big data, genetic EHR, and imaging. Degui.
Hi, good morning. I'm really excited to come back to UAB because I was there for 7 years in my tenure track, and I'm glad to see a lot of old friends and new friends. And also thanks Mark for a kind introduction. It brings me back to even further my PhD years. So I'm just today going to share what I do in the past few years. Mostly, I mean, I make the groundier title. Really, what I'm talking about is say how can we just maybe a little bit philosophical question. What the data science for big data really do? Right? I mean, we all this informaticians conference. I think I mean we are not only moving around big data. I mean, it's faster and a bigger chunk. But what's really important is we hopefully bring new insight into the problem, not detectable in small data. So I'm going to show my lab's experience in 3 domains: genetic big data, EHR big data, and imaging big data. It will be a quick overview. Sorry, I cannot go into too much detail on each of those. So talking about genetic big data, really, just a few years ago, we are looking at smaller cohorts, individual epidemiological cohorts, only tens or tens of thousands of samples. And we are happy, right, doing a lot of GWAS published everywhere. But now, really, if you look at the field of GWAS, I mean, it's all dominated by biobanks, UK Biobank especially. And all of us, you know, it's coming. It's already released the first batch of genetic data a few weeks ago. And in that domain, doing GWAS is great, but I think there's more opportunities beyond the single variant association. Mark mentioned the structure variation, but I was especially excited about the long haplotypes or shared DNA segments called IBDs. And moving toward in the future, I guess our genetic data is only getting better, bigger, and I hope we introduce you this possibility of doing something different. So quickly, I'm talking about the IBD segments or identity by descent. Those are the segments inherited from common ancestor. This is a small family, but looking at the bigger population, it's the same. So we're going to share a lot of those things. You don't see that when you have small data because families are sampled very sparsely. Now we have a bigger sample closer to the population size. I mean, in 2018, it's shown that even sample 5% of the entire population, you are guaranteed to find some of your fourth degree cousin or third degree cousin. And what we are analyzing the data even on the smaller slice, namely UK Biobank, covering 0.7% of the entire UK population. We're going to see tremendous coverage on each and every chromosome. Basically, 80% of the chromosome will be covered by some shared IBD segments over 5 centimeters. I mean, just in population on average. So that's really bring us a lot of dense coverage of the entire genome. And what can we do, right? So one obvious thing is to genetic genealogy and can trace distant relatives. But in my lab, we have developed a fast method called a RAPID, which can identify those segments in near linear time of the sample size. Note that traditionally it is mostly quadratic. And what we do, we not only find those segments, but if you post-process those segments, you can find genetic relatives. So previously, genetic relatives are called by a method called King. It's more than 10 years old. It's great method and it's still being widely used nowadays, but it won't scale up much further beyond the UK Biobank sized cohort. Mainly just it's doing pairwise comparison. So we developed the method called RAFI or RAPID, our IBD methods for accurate and fast family inference. And we are up to 10 times faster than RAFI, than King, and also maintain comparable, even better, robust and accuracy. So another sample of our work is when we calling those runs of homozygosity segments out. So you know the runs of homozygosity is a thing, and it's there, but people don't analyze it that much, mainly because we don't have a tools to call them. You can use a Plink to call individual RH from each one, but you don't know the consensus. And then typically people just aggregates the total RH content in the genome and lump together and compare case and controls. what we do is we use the efficient data structure, PVWT, and that's the same data structure we leverage for our rapid methods. And we call them segments, again, near linear time and very fast. And so that we can call those consensus runs of homogeneity segment out directly. And then we can enable association in the loci specific fashion. Indeed, for example, at the beginning of the pandemic, we analyzed the UK Biobank COVID-19 data. We find one haplotype or one diplotype, RH, in chromosome 4 near the PPA2 gene that's associated with COVID-19 related deaths. And it's plausible because PPA2 gene was previously associated with a sudden cardiac death in infants. So it's possible. I mean, it could be related to cardiac related deaths. Okay, and moving forward, I'm going to talk about the electronic health record data. It's a very different domain. I started work after I joined at Houston. So in our school, we have access to large EHR data from commercial vendors, and they are really much bigger than even the UK Biobank. They're mostly containing the patient's structured EHR data, so-called, and mostly de-identified, so enabling to some mostly population health-based analysis. But we are thinking maybe we can build some predictive models. So in 2018, my student Lela show that using the Sonar Health Fact data, and we can define a much larger cohorts than anybody in the literature. We have about over 1 million patients, and in just heart failure, right? You won't get in individual hospital, and then the benefit is that when we putting the data together, then we can build a deep learning model. We show that the model, if it's trained on all the data, it will perform and then fine tuning on individuals small hospital. It will perform much better than the model just trained on the hospital data by itself. Right? So that's the benefit of using big EHR data. And from there, you know, the field of deep learning moving fast. They have BERT model and a whole smear of large language model, and people call it foundation model. Basically, the idea is that you have mostly text, but also now into images. You use a large foundation model absorbing the data, and then using that model, you can enable a lot of downstream tasks. And in practice, oftentimes you only need a very good foundation model and then fine tune for your own purposes. So this is sort of quick architecture, but it's mostly a transformer, right? And what we do say, instead of in the NLP or text domain, we can feed the data, the structural EHR data directly into the transformer. And we can predict any disease risk that we want. And of course, I need some engineering to change the EHR data modality, make it look like a natural language sentences, and to fine tune this the architecture but we eventually make it working, and we show that when you have MedBird pre-trained, we enables the predictive modeling performance being boosted by pretty large margin. And is that also equivalent to almost increasing the sample size by for the factor of 10? That's how it could be potentially very useful for training your local model. And then just yesterday, literally, this paper is out. We show using even larger data from Sonar real world, and we train a COVID-19 prediction model, and then it can effectively stratify patients and hopefully with can be deployable into practice. So lastly, I just want to quickly show our work also in imaging big data. You know, imaging has been powered deep learning developments since early days, and also has success in medical domain. And UK Biobank, luckily, they released their imaging data, and it powered some initial UK Biobank imaging-based GWAS papers. So, but it is when we see it, it's most these studies are using phenotype that defined by expert or heuristic. They are sort of non-deep learning based. It is fine. They find a lot of signals, but it will miss a lot of other information that is not easily articulated by human. By the experts, and especially 3D images. You can't imagine we are trained to read the 3D brain images as human. So people develop supervised machine learning methods, deep learning, and can just predict those phenotype defined by human, but it requires human labeling and then still carry the same limitation. So what we did, Chen lab is we think maybe we can define some descriptors from images, not by human labeling. We'll use so-called self-supervised learning. And the idea is that when, say, this is the retina fundus images, we train a neural network such that it will output those vectors and phenotypes from the individual images such that the same person's images will cluster together and different people will be far apart. So that's called a contrastive learning. You don't need any label. All you need to just feed a lot of eyeballs into the model. It will learn. And similarly, we recently trained a model for 3D images, 3D MRI from UK Biobank. We show that, we don't have one brain, it's difficult to do the contrastive learning, but what we do is through this autoencoder and then to reconstruct the input modality. And these are real images. You can see the reconstruction sort of makes sense. It didn't capture all the details, but captured the overall shape pretty well. And when we do this, we know this is not capturing all information in the image, but it will carry some inherent information, right? They probably good enough. So here I'm just quickly show because since people love to show UMAP today, those are individual. Each dot is one of the individual's brain image, and we show after training the brain image correlates those embedding correlates with usual covariates, sex and ages, but also the brain related like a white matter volume. I just show example, but different measures also have pretty good correlation in both images embedding from T2 or T1. And using those phenotypes, we conducted GWAS, genetic association studies, And in the retina, we find some 34 loci, and imaging we find even more. So we are excited to wrapping this up. So just in conclusion, we hope to show very quickly that using big data, we can enable the discoveries that is not possible in small data. And I really, all the heavy work done in my awesome lab, and beyond my lab, current member and past members, there are collaborators everywhere. That's really make us much powerful than our alone. And also the funding agencies. Thank you. I can take some question. I don't know if I have time.
Yeah, thank you, Degui. We have time for a couple questions.
Degui, congratulations on your new accepted paper in Lancet subjournal. That's awesome. So a naive question. I'm not so we are working, not me, but Dr. James Cimino, and we have a few folks working on clinical informatics or EHR information. So it's pretty dirty, right? How did you like harmonize or try to clean the data, especially some text, clinical text information, or if you select some like well characterized traits or clinical information?
Yeah, really good question. I think it's philosophical too. I mean, some people love clean data, and I kind of love messy data. I mean, the messy data, I mean, you'll be more careful. So deep learning is that it's large model. It can pick up weak correlations, and you don't rely on individual signals too much. So I mean, for example, the success of MedBird, we have the MedBird is very large model, and we don't like to predict individuals, say heart failure super well, just focus on that. We want to train a more general model that absorb the information from the data, and then for any downstream task, you can fine tune with maybe you clean the data or any specific like domain knowledge you can inject. So that's one way of dealing with the messiness, but of course there are other ways. And then by the way, we are working with structured EHR, which is the non-text part, with clinical notes and some. They're very useful, informative, but it's not part of this work.
Great, thank you.